Okay, so we're gonna go through how to do the problems for the Mendelian genetics. Okay. So the first question is asking about what is the probability of having 3 girls in a row in one family? So every time there is a baby being born, there is a 50% chance or a 1/2 chance of it being boy or a girl, okay? And when you're trying to calculate probabilities for compound events, which means that, we're saying that we get a girl one time, and then another 50% time for the second girl, and another 50% time for the third girl. It's a compound event, so what happens is you just multiply these together, okay? And so the answer is 1/8, because you do 2 times 2 times 2, so it's 1/8. So this is the probability of having 3 girls in a row. It'd be the same thing having 3 boys in a row. And then if you want to add an additional girl, you'd have to do, it's 50-50 chance for the first girl, 50-50 chance for the second girl, 50-50 chance for the third girl, and then a 50-50 chance for the fourth girl. Okay, so it doesn't change the probability. Having a girl does not affect what child you'll have next. It's always a 50-50 chance. And so for this one, it would be 1 out of 16. Okay, so again, you just multiply these together. So 1/2 times 1/2 times 1/2 times 1/2. And since these are all like ones on the top, those just stay as ones. And you really just multiply the bottom. Okay, so this is kind of the law of multiplication and has to do with probability. Okay, so that is the first problem. The second problem has to do with brown and blue eyes. Okay, and so what happens is brown eyes are going to be dominant. So capital B is going to be brown, and lowercase b is going to be blue. Okay, and we know that brown is dominant over blue. So then we can say that when we have big B, that's going to be brown. We have big B, that's going to be brown. And when we have little b, that's going to be blue. Okay, so anytime that they say something like, that brown is dominant over blue, then in the heterozygote form, that brown is going to be the color of the eye. Okay, so the question says, a brown-eyed man marries a blue-eyed woman, and they have 3 children, 2 of whom are brown-eyed and 1 of whom is blue-eyed. Draw the Punnet square that illustrates this marriage. What is the man's genotype? What are the genotypes of the children? So we have a brown-eyed man. So the way I'm going to write this is he's big B blank. We do not know what this is, okay? And he's going to marry a woman that has blue eyes. So we know that she has to be little b. When they have children, 2 of the children are brown eye. So 2 kids are brown, and one child, our kid, is blue. So this child right here tells you what the genotype of the man is. In order to get a blue-eyed child, you have to have getting a little b from both of the parents, okay? And so what the genotype of the father is going to be is he will be big B, little b. The mom will be little b, because we know that she's blue-eyed. And then we can do a Punnet square showing the cross between these 2. Okay, and when you're trying to figure out what are the letters that I put here, for these ones, you just put the individual letters, okay? And you just have to separate them from each other. So we have the father up here, big B, mother, little b, and then we can do our cross. Okay, and so this is going to be what the children could be. You could have children that are going to be big B, and little b, okay? So for this one, it says, what is the man's genotype? So the man's genotype is going to be this. And then said, what are the genotypes of the children? These are the genotypes of the children, okay? They are always going to get a little b from their mother, so they have to have a little b there. Okay. moving on. All right, so in this next question, it's about dogs, and there's hereditary deafness. It's recessive, okay? So, and they're using the letter D. So we're gonna write out D. So D is gonna be a normal hearing dog, capital D. Okay, and then it says, deafness is caused by recessive gene little d. So little d is gonna be deafness, or deaf. We have a kennel owner that has a male dog that she wants to use for breeding purposes. The dog can hear, so we know that the dog has at least one big D. We don't know what the other thing is. We know that the dog can hear, so the owner knows that it's either going to be, it could be, so if a hearing dog is going to be big D, or big D, little d. So this is hearing. In order to be in deaf, you have to have both of the little ones. So, all right. However, they don't know what the dog's genotype is, okay? If the dog has a little d, they're not gonna want to breed him because there's a higher probability of him creating offspring that have deafness. So you'd really wanna have a homozygous dominant male for breeding purposes, okay? And so in order to figure out what this male's genotype is, you can do some crosses, okay? And so what does it say? The owner does not wish to use some breeding, so the deafness gene will not be passed on. This can be tested by breeding the dog to a deaf female. So deaf female would be the little d. Draw the Punnet squares to illustrate these two possible crosses. So the first cross would be a big D, times this. And the second cross would be if he did carry the deafness gene, this one. And so what we can do is two Punnet squares. Okay, and so this would be big D, little d. This one over here would be big D, little d. And so we can go through. Okay, and then this one. Okay, so this really shows you why you'd want to have a homozygous dominant male for this cross, because none of these would be deaf. So 0% are deaf. If the male is in fact a carrier for deafness, then 50% or 1/2 would be deaf. Okay, so this is the kind of male you would not want to use in this cross. However, unless you do an actual genetic test, the only way to do this would be to do a test cross. And so a test cross is when you take an unknown genotypic individual, they have the dominant genotype, but you don't know what their other gene is, and you cross them with a homozygous recessive, and then that shows you if they are, in fact, a heterozygote. Okay, and so this individual would not want to be used as a father for other puppies. Okay. And then it says, how could you tell the genotype of this male dog? You're gonna tell by the offspring. Okay, so you look at the offspring. If you have deaf puppies, then you know that the male has that heterozygote situation. Okay, and then the second part of this says, what would happen, show how two hearing dogs could produce deaf offspring. Okay. And in this case, what's going to happen is that in order for two hearing dogs to produce deaf offspring, you're going to have they have to carry the gene. So they're both going to be heterozygotes, basically. Okay, so you have to have heterozygote. That means that they both carry a gene that is for deafness. Okay, because the only way to make a deaf offspring is going to be to have this little d. Okay, and so we call these carriers, will produce most offspring hearing, but they will be able to produce a deaf offspring right there. Okay. All right, next question. So in this question, we have grasshoppers, and the grasshopper is black with red and yellow stripes. And we assume that the red stripes are expressed by G to the R. So this means that the grasshopper is going to have red stripes. And yellow stripes are going to be G to the Y. And have no stripes, you're going to have a little G. Okay, the second that you start seeing something written out like this, you need to start thinking codominance, okay? Because this is an interesting situation where you have, first of all, 3 different potential phenotypes. And when you start kind of writing the G and then the to the R and G to the Y, that makes me always think that this is going to be a codominant situation. Okay, so when we look at this, it says what will be the phenotypic ratio resulting from a cross of 2 grasshoppers, both with red and yellow stripes. So if we have grasshoppers with both red and yellow, this is what their genotype is going to be. Okay, and so then we can do our cross. Okay, and then you basically just separate these. This would be G to the R. This is G to the Y, G to the R, G to the Y. Okay, and so this individual is going to have just red stripes. This individual is going to have both red and yellow. This one is going to have red and yellow. Red, yellow. And this one is going to have just yellow. Okay, and so this is the genotypic ratio. So we're going to do the genotypic ratio. We basically just have to figure out, okay, so we have one big G like this. We have 2 of these ones right here. So there's GR, GY, okay, so 2 of them. And then there's 1 GY, GY. Okay, so this is the genotypic ratio. Okay, because genotypic ratio is the genes. Now, the question asks for the phenotypic ratio, so you have to make sure you do this. This is then going to be, what does this actually look like? Okay, and so this is going to be red stripes. Red stripes. This is going to have red and yellow. And then this one's going to be yellow only. Okay, and there's 1 of these, there's 2 of these, and 1 of those. And so the way that you can write this is 1, we'll just say red, not plus sign. You just do dot, dot, which means 2. 1 red to 2 red yellows to 1 yellow. Okay, and so that would be the ratio, or you could leave it like this, but you want to make sure you add the numbers because more of them are going to probability be this red-yellow combination because there's 2 ways to make that combination. Okay, there's only one way to make these 2, so this is going to be the kind of the bigger type. Okay, and then it says what type of inheritance is this situation? So if both of these, it didn't say anything about there being kind of like an orange stripe. If they said that if red and yellow mix, then you get orange, then we'd be like, okay, this is incomplete dominance. But in this case, both of them continue to show up, and so this is going to be co-dominance. Okay? All right. Okay, this next question is about this chemical test that you can do. We normally do this in lab, but obviously we can't do it now. But it's called PTC, and it just stands for this long chemical name. But basically, some people can taste it, some people can't. I actually can't taste it. So the way that you do the test is you take a little piece of paper and you put it on your tongue. And for people that can't taste it, it just tastes like you have paper in your mouth. For people that can taste it, it comes off and it tastes very bitter. And so there is, most people can taste it. So it's the dominant allele. So being a taster, capital T, is the dominant situation. Okay. And then for us that can't taste it, we are non-tasters. So we're going to be the recessive allele, non-taster. Oops, that says tester. All right. Okay, and because we're talking about dominance in this situation, this is going to be a complete dominant situation where we have big T, and big T, little t are going to be tasters. And then little t is going to be the non-taster. Okay, so that's the first part. Then there's the second part that talks about albinism, which is being albino. Okay, so people that lack pigment in their skin. So albinism is also a single locus trait, and normal pigment is dominant. So this is going to be normal pigment. And the recessive is going to be albino. Okay, and this is a normal dominant trait, so it's big A, and big A, little a will be kind of normal pigment. And little a is going to be albino. Okay, so I always like to do this first to figure out what's going on, okay? Because then I know that if I start seeing these things together, I know what it means. All right, so now it says a normally pigmented woman who cannot taste PTC. So she's normally pigmented, so we have this A's. It does not tell us anything about this letter right now, okay? So let's just do that. So normally pigmented, so we know that she at least has to have one capital A's. She cannot taste. So the only way to have a non-taster is to be little t. Okay, she is then going to have a baby. Oh wait, sorry. It also says that this woman had a father that is albino. So in order for her father to be albino, he has to be big A or little a, which means that he had to have given his daughter a little a. Okay, so we know that she is a carrier for albinism. Okay, then she marries a homozygous, normally pigmented man. So homozygous, normally pigmented, which would mean this one right here. So homozygous means they have to be the same. Man who is a taster, but who has a mother that does not taste. And so when they're saying that, they're basically telling you that this person is a heterozygote. Because if their parent is a non-taster, the only allele that she could give to her child is a little t. And so you know that he has to be a little t. And that same situation for the woman. Her father was albino, which means that she has to have gotten a little a from her father, because that's the only allele type that he has. Okay. So then it says, what are the genotypes of the possible children? So this is a dihybrid cross, okay? And so I'm going to write this bigger in a second. But first, what I want to show is how do you get the different things for the top, the different allele combinations. And so you basically have to say, well, I'm going to have a big A with a little t. There's actually going to be two of those because it could be this A with this t and then this A with this t. So I write it twice. And then there's going to be this A with this t, so that's little a, little t, and this A with this little t. Okay? For this one, we're going to have this A with this t, and we also could have this A with this t. And then we can have this A with this little t, and this A with this little t. Okay, I know that they're identical. I'm just writing them out to show you that, because if I had these in different colors, because technically these are on two different chromosomes. So the way that they can line up is, that's independent assortment. So you have to show that they can line up in any of these ways. So these are going to be our possible sperm and egg combinations, okay? So let me just draw out my box. Okay, and this one has to have more boxes because it's going to be a dihybrid. Okay, then I go in and I'm going to take all of my female gametes and I'm going to write them on top. So this is big A, little t. Okay, and then I'm going to take my male ones right here. Okay, and then what's going to happen is you go through and then you have to write out these. So I'm going to not say these out loud because I tend to mess up when I start saying them out loud. So I'm just going to write them. Okay, so I've written those out, and then it's asking for what are the genotypes of the possible children. So I basically think you could just leave them in here, but you can also write them kind of out. It just makes it, this is a little difficult because you have to then figure out which ones kind of go where. So you could like add them up, because some of these are the same, like AA, capital A, big A, little t. Those two are the same. But I think leaving them in this format is completely fine. So these are the genotypes, okay? And it doesn't say the ratio of the genotypes, which makes me say that you can probably just leave it like this, okay? If it said the ratio, then you'd have to be like, all right, how many of this type do I have? I have 1, 2, 3, I think those 3 of that kind, and then you'd have to count through. So that is one way that you'd have to do this. This is unlikely to show up on like a quiz or a practical, kind of having to do the full dihybrid cross, because it's a lot. Okay, but then looking at this, we can then say, well, what percentage of these children are albino? And so the only way to be albino is you have to have little a, and none of these have little a. Okay, so I can't see any that are little a. So for that one question, we're going to say 0% are albino. Okay, and then it says what percentage of children will be non-tasters? Okay, so they're going to be the homozygous recessive. So there are some of these. So I'm going to circle them. So this is one. Actually, all of these. Oh no, that's not one. This is one. This is one. Basically, the half of the chart is, these ones all have capital T, so they can't be non-tasters. And so because this is 8 out of 16, this is just 1/2. So 50% are going to be non-tasters. Okay, and again, I'm just looking at just the T's on this one. Okay, so in order to be a non-taster, you have to have little t. Okay, so that is how you're going to do that problem. All right, the next one. Okay, so it says, suppose you have 2 rose plants, both with pink flowers, okay, and you cross the 2 plants and are surprised to find that while most of the offspring are pink, some are red and some are white. You decide that you would like red flowers and would like to make more. What cross would you perform to produce the most red flowered plants? What type of inheritance is this? Okay, and so what I'm going to do right now is, it doesn't tell me what letters I'm using, but I'm going to make them up. So I'm going to say that capital R is red, and lowercase r is going to be white. Okay, but this is, you're seeing pink show up. So what's going to happen is big R, little r is going to be pink. Okay, and I guess I should just put these with their full genotype. Okay, so in this situation, right away, I want you to think incomplete dominance. Okay, because anytime that there's a blending, this is like blending paint. If you have red paint and you have white paint, you are going to make pink, okay? And so the question then asks, what is the, what cross would you perform to produce the most red flowered plants? Okay, and so if you have, the one to make the most red is if you take 2 reds and you cross them, then it's going to be this. Okay, so you would have all red. Now, the way that I usually answer this question is that, well, you have, okay, so you've done a cross, you have, the first cross would have been this big R, little r, big R, little r. And you would have had, you mostly have pink flowers. And so another cross that you could do, so if you don't have enough red flowers to do this, you would want to do a red flower with a pink flower, because this would also produce more red flowers than if you would do like 2 of the pinks together, because then you would get this. Okay, so I find both of these answers acceptable. It just kind of depends on what number of like if we're gonna say that you have to use one of the pink flowers to make your cross, then this could be a possible answer, because then you'd have, in this case, 50%. Obviously, if you want just red flowers and you had enough red flowers to make a cross of just red, then you would have 100% red right here, okay? But it just kind of depends on what you're allowed to do. And then from the question, it doesn't really say what you're allowed to do. So if you want to be safe, you would just write this one right here. For individuals that are in my class, if you wrote this, I would give you all the points. But this is really important to write. So this is incomplete dominance because you have a blending of the two colors. Okay, so moving on. We're now going to talk about hemophilia. And so hemophilia is a sex-linked trait. So you have to use X's and Y's in this case. You can't just use just the H, because we're going to use, I guess it doesn't give you any letters, but we'll use H. So it says hemophilia is a recessive sex-linked trait. So first of all, I'm just going to kind of put this out here. So we have capital H is going to be non-hemophiliac, non-hemo, and we're going to say that this one is a hemophiliac. Hemophilia has to do with kind of bleeding too much. You bleed excessively. Okay, but this is a sex-linked trait. So what that means is that this is going to be found on the X chromosome, but not on the Y chromosome. So when you write things, you're going to be like, you can write it like this. So it's going to be, for females, you can be capital H, you can be this, or you can be this. Okay, this is going to be normal. This is going to be normal. This is going to be a hemophiliac. This person is also, we can call them a carrier because they carry the gene. Okay, for men, it's a little bit less complicated because it's only going to be on the X chromosome. So the Y does not carry a letter. So it's going to be just the Y. So this is going to be a normal. And if the person has the hemophilia, they're going to have hemophilia. Okay, so there is no heterozygote combination in this case because they only have a Y, and the Y does not carry this trait, okay? So then it tells us that a non-hemophiliac man, and so for a man to have non-hemophilia, this is gonna be the only thing he could be right here. So he's gonna be with a capital H and a Y. Okay, so that's what we know. And then it says that a non-hemophiliac man and woman marry. So the woman, so she is going to, we don't really know what this other letter is. We know that she's not hemophiliac, okay? So she at least has to have one of the capital H's. And it says that they have a daughter who's a hemophiliac, okay? So then they have to produce a child that has this. So this is the only way for a child to be hemophiliac. The father thinks that the woman cheated on him. Okay, and the question is like, does he have evidence that this, is there evidence that this is true? And there is, okay? Because what we can say is that, yes, she could have had a little h, and she most definitely has to have a little h. But in order to produce a female child, okay, if it was a male child, he would not know at all. Like, because then we would just assume that she was a carrier, and that's, he gave the Y and then she gave that, and that's how it would happen. So we would not think there's cheating. But because it's a female, the only way to get a female that's a hemophiliac is you have to have the faulty gene from both the father and the mother, because the father is giving the X chromosome to his daughter. That's the male is the one that determines the sex of the child. And so since he does not have hemophilia, there is no possible way that he could have a daughter with hemophilia, okay? And so most definitely there was cheating. So yes, there is evidence that cheating took place, okay? Because this is what we would assume is the cross right here. This would not produce any female children that were hemophiliacs. And I'm just gonna show you what that would look like so we can actually see the cross out. Okay, so this is gonna be the father's, this is the you know, the man who's married to the woman. We'll just do that cross so you can see that this would be impossible for there to be a female child that is hemophiliac. Okay. So same thing, you do XH, we have the Y right there. This would be X capital H, X lowercase h. So this would be a female non-hemophiliac. This would be a female carrier. This would be a male non-hemophiliac, and this would be a male hemophiliac. Okay, and so there's only one possible child that would be hemophiliac. There's no way to make a daughter, okay? And so because the father always donates X's to his daughter, he would have to be a hemophiliac for that to actually work. Okay, so unfortunately, yes, there was cheating in this relationship. Okay. All right, so blood typing is a little bit more confusing. So this is the next one. And first I want to just write what the blood types could be. So you can have A, B, and O. And then I'm going to write what the genotypes would look like. Okay, and we always use I's. So this could be, to have a blood type, you could be this or like this. Okay, so this little is like a recessive. For B, it could be this or this, okay? There's only one possible way to get an AB, would be like that. You can't have another I or anything. And the only one way to be O is to have two little I's, okay? So it says a woman with type A blood, okay, so she's gonna have type A blood. So we know that she's this. We don't know what this is right here. It says her daughter has type O blood. Okay, so this woman is going to produce a child that has this. Okay, the only way for her to produce a child to do that is that she would have to have a little I, because she's going to give these to her children, one or the other. This would not produce a child with little I, so she has to be a carrier for that right there. Okay, and then it says, what are the possible genotypes and blood types of the child's father? So basically, the father just has to have at least one little I, but anything could be right here. So this could be, he could be this, he could be this, or he could be this. Okay, so these are the three possible genotypes that could produce a child that has this. Okay, this is not sex-linked, so it doesn't matter. We're not thinking about X's and Y's at this point. This is on another chromosome. But the only way for a baby to be produced with little I is that the father and the mother both have to have a little I. Okay? All right. This last question is about color blindness. And for color blindness is recessive, okay? But this is also going to be a sex-linked trait. So it's the colorblind gene is located on the X chromosome, and there's nothing on the Y chromosome that corresponds, okay? And so for this, we're gonna say that capital B is sighted. So I guess I shouldn't say sighted because these people aren't blind. This is, so normal color vision. And little b is gonna be colorblind. Okay, and so this is recessive, so only the little b little situation is going to be colorblind. Okay, so I'll just put colorblind. Okay, both of these are going to be normal. All right, so it says, if a man and a woman, both with normal vision, marry and have a colorblind son. Okay, so in order for this to happen, you have to have normal vision. So I'm just going to write this out because this is a sex-linked trait, so XY. Okay, so if a man has normal vision, that means he has to have a capital B. Okay, because there's nothing on here, and if he has normal vision, he has this gene. Okay, this woman also has normal vision, but it's telling us that they have produced a child that is colorblind, so we know that she has to be the one that's carrying it. Okay, because she doesn't have it, but she has the allele. Okay, So both with normal vision, marry and have a colorblind son. Draw the Punnet square that illustrates this. Okay, so this is what it would look like, and then we can draw the Punnet square. I feel like I drew this too small. Okay, so okay, and here's our colorblind son. Okay, so this is again a very good example of why males are affected by certain sex-linked traits at a higher rate, because this female right here, she also got this faulty gene, but because she got the normal one from her dad, she's normal vision. So only the male child would be the one that has the color blindness. Okay. Then it says that if the man dies and the woman remarries to a colorblind man, okay, so this, we're kind of getting rid of this guy. This woman is still the same. She's not, her genotype has not changed. So we're going to take that. So she's X, capital B, X, little b. So she marries a man that is colorblind. Okay, so then that means that he has to have a colorblind allele, and that's all he has. Okay, and then it says, draw a Punnet square, and then look at the phenotypic ratio. Okay, so we know that she's a carrier, and now that we have a father that is colorblind, we're gonna have an increased number of colorblind offspring. And most likely a girl is also gonna be colorblind. Okay, oops, yeah, so. So in this case, neglecting to look at actual sex, you could just say that half of the children, so these ones right here, are going to be colorblind. So half are colorblind and half are normal vision. Okay, so that would be then how you would do these questions. Okay, if you're having a hard time doing like typing X to the B, just print these out, do them by hand, and just send me pictures of them. Okay?